everyone, this is Amanda Moore from the Angelman Syndrome Foundation, and you're listening to the ASF Podcast, the show that explains everything you want to know about Angelman Syndrome. We're so excited you're here, and we hope you enjoy the show.
Hey, everybody. Well, thanks for joining us on the Angelman Syndrome Foundation podcast. My name is Christian Hommes, and I'm going to be your host for this episode. And I'm joined today by Matt Judson. Matt works down at UNC. So, Matt, thanks for joining us and welcome to the podcast. How you doing?
I'm doing pretty well. Yourself?
Not too bad. Not too bad. Well, before we get started, just tell us a little bit about yourself. So where are you from originally? What your background is, and how did you get involved with Angelman research at all?
Yeah, no problem. So I'm from St. Louis, Missouri originally. Grew up there, went to undergrad in a cornfield right on the Missouri-Iowa border, Truman State University. And it's where I met my wife, and we've been married for a good while now, got 2 kids, and just enjoy a lot of kind of leisure activities besides research. So just get out, do a lot of distance running when I can. And North Carolina's got just a lot of things that Missouri doesn't in the way of beaches and mountains. So I love to do some visiting out there and do a little leisure reading that's, not scientifically oriented. So that's always a good release.
Yeah, so you're not always focusing on Angelman mice in that regard?
Not always.
That's good.
There's a sliver of time away from the mouse colony sometimes.
Yeah. So how did you end up at UNC?
Yeah, it's a great question. So I was always been interested in research, and I kind of knew I wanted to pursue that in graduate school, but I didn't really even have a crystallized neuroscience research per se until kind of a little bit into my first year at graduate school. And then I just kind of got swept up in a really awesome autism research lab, Pat Levitt's lab. He's out at University of Southern California now. And kind of was studying genetic factors underlying idiopathic autisms. And so when I was looking for postdoctoral fellowships, other neuroscience labs like the Philpot Lab, where I'm at now, were very attractive. And at that time, Ben was just getting into Angelman syndrome research. So it's kind of interesting. I went out for my interview out there kind of expecting to do things more tangentially related to autism research, like, kind of experience-dependent plasticity research, things like that, really hardcore synaptic physiology. And all Ben wanted to talk about was Angelman syndrome research, which I was familiar with because we had a strength in that area at Vanderbilt, where I was at grad school at the time in Pat's lab. But yeah, and kind of the rest is history. So I've just been really, swept up in this vein of research and a lot of, UBE3A related neurobiology for a little over a decade now.
Oh, wow.
Yeah, so I did my postdoc here, and I'm a research assistant professor here at UNC, and just got a lot of good active research in the field.
Yeah, how many people are in your lab right now?
I think it's about 16 to 18.
Wow.
We have a kind of a flux of rotation students and undergrads and things at times, but yeah, kind of our core grad student, postdoc, and research associate kind of core is around maybe 10 to 12. So we've got a pretty good, strong team. We're getting a lot done in a lot of, various kind of domains of UBE3A research right now.
Nice. Well, we're going to get into your research at the end a little bit.
Sure.
But yeah, so my idea for this episode is specifically about UBE3A. And I think that it's hard to find a really concise platform that talks about it. There's lots of really intricate lectures and videos you can sift through to find some information, but I don't know if there's really, a quick and concise talk about it that breaks it down for either the layperson like myself or even some clinicians out there. And I find that every year I'm kind of researching it myself to understand it better, just because in order to understand the new therapeutics, I've got to do a refresher and, understand the new data and research that's out there. So hopefully this will just be kind of a refresher for a lot of parents or maybe new information to. But yeah, I think you're the perfect person for it. I think you break down scientific information well, and I've enjoyed the lectures at the ASF conference you gave. So yeah, I'm excited
Great.
that you're
I'll
joining me.
do my best. That's for sure.
Right. Yeah, well, we can get into it. So we know that Angelman syndrome is caused by a lack of UBE3A or a fully functioning UBE3A. And why is that gene so important? What does it make? What does it do?
So the UBE3A gene, which is, kind of the proximal molecular cause or loss thereof of Angelman syndrome, it makes a really special protein called UBE3A. It's an E3 ubiquitin ligase. And just in lay terms, what it's really doing is it's a special kind of enzyme that works with some other enzymes to tag a cell's unwanted proteins. So a cell's always kind of, making new proteins and getting rid of old ones. There's this kind of homeostasis it's got to keep up, right? So some housekeeping. And it can tag these kind of unwanted, sort of used up, misfolded proteins maybe with a tag called ubiquitin. It's another protein. And if you can kind of do this a number of times, you get this chain of ubiquitins. And this is sort of a signal to the cell that says, all right, this unwanted protein is ready to be degraded and turned over and kind of chewed up into its kind of component amino acids. So every protein is made up of little amino acids. And the thing that chews it up upon this signal being recognized is the proteasome. So UB3A works with these other enzymes in the proteasomes to get rid of these unwanted proteins.
Okay, gotcha. So it's cleaning things up mostly in the synapse, just all these different waste products, things like that.
So UB3A is enriched in synaptic compartments of a cell or a neuron. It's also really enriched in other compartments of the cell, like the nucleus. So it could be turning over proteins, excessive proteins, unwanted proteins, and all sorts of different compartments of the neuron.
Gotcha. Okay. And there's probably, do we have any rough idea of how many chemicals or waste products it targets? Or is it just too many that we can't even fathom at this point?
I think that's the $1 million question right now is, what are all of the proteins that it might interact with and target for degradation by the proteasome? And moreover, which of those are most critical and at which kind of points in neurodevelopment? And the short answer is we don't really know the answer to that question. So there's definitely very interesting proteins that are known to be targeted by UBE3A for degradation. One of them is a synaptic protein, just for example, called effexin-5. And it's known that effexin-5 is really important for excitatory synapse development. But just kind of as a caveat, we also know that effexin-5 is really enriched in the hippocampus. And so, even if, effexin-5 is the most important UBE3A substrate for degradation in the hippocampus, it still, begs the question, all right, what about other parts of the brain? And what about other, specific points along the neurodevelopmental trajectory? So, we've got space and time in the brain to consider, and also potentially a number of substrates. So the short answer is we don't really know how many, and we certainly don't know what all they are at this point.
Gotcha. So it's not necessarily there's one key waste product that is so high that causes the majority of symptoms or issues with Angelman. It could be just a multitude of factors there.
Yeah, it doesn't seem to be the case. And also it may not be things that are strictly speaking, waste products. There may be certain proteins that have to be maintained in a really tight range in terms of their levels for optimal function of the cell. So like something that's, you think of like a rheostat, you just got to get just the right level, and UB3A might help tune that in through protein degradation.
Okay. And so does that relate? So if there's a buildup of these products, is it harder for the neurons to talk to each other then?
It could be, yes. Especially if you imagine a synaptic protein that is kind of like, something like I just mentioned, effexin-5, sure, that could affect the number of synapses that a neuron makes or develops. And so that obviously would change the quality of communication that a cell can have with other cells in a circuit. If you think of maybe UBE3A in the nucleus, it might be involved with turning over transcription factors that in turn regulate the expression of a lot of genes, like a whole kind of network of genes. So the effects could be really far reaching. And it's kind of important to think about that. And it's really one of the challenges for the field to try to understand what happens downstream of UBE3A loss.
Yeah, lots of downstream effects that could be there. So the history of UBE3A, how did we even come up with this, or why did we look into this gene to begin with? How did we discover that it was really the issue with Angelman?
Yeah, so to my understanding, it was kind of this fascinating kind of clinical observations that seemed sort of paradoxical back probably in the 80s, where people were noticing, clinicians were noticing, and affected families were getting these diagnoses of like, hey, there's this region of chromosome 15, this 15q11, q13 region that's being deleted. And sometimes the deletion was being associated with Angelman syndrome, and sometimes it was being associated with Prader-Willi syndrome. And it's like really kind of confusing, I think, at the outset, probably. I mean, I'm speculating a little bit there, but yeah. I think what sort of made it, a little bit clearer was the recognition that, hey, it's when it's the paternally inherited chromosomes deleted. That's when it's a Prader-Willi situation and an outcome. And then when conversely, when it's the maternal deletion of the same region, that's when you typically have an Angelman syndrome outcome. And then I think from there's recognition that there's kind of methylation or like a mechanism for imprinting this region. And it turns out that if you have there's paternally imprinted genes there's a paternally imprinted gene UBE3A that's really only expressed from the maternal chromosome in neurons. And then in later studies, it became clearer that, okay, UBE3A is really the key gene in this 15q region that's being lost in Angelman syndrome.
Gotcha. Just kind of trial and error and figuring out what genes, are lacking or present in different patients. I gotcha.
Yeah. So by the 90s and early 2000s, it was pretty clear that, okay, UBE3A is in neurons is really only being expressed off of that maternal allele. And then, there was identified, circumscribed deletions that just included UBE3A or point mutations of the UBE3A enzyme itself. And that was also yielding Angelman syndrome diagnoses. So it just became clearer and clearer that, okay, UBE3A really is this focal point on maternal 15q for Angelman syndrome.
Okay. Yeah, I think that's been probably one of the most fascinating things that I learned as a new parent, learning about Angelman syndrome, is that this on the paternal chromosome UBE3A is silenced only in the brain, though, correct? So in other cells in the body, it's not there. That antisense is not there.
Yeah. As far as I know, there could be peripheral nervous system imprinting too,
okay,
but
gotcha.
I don't know of other tissues besides brain tissue, specifically neuron.
Do we have any theory of why that would be the case? I know it's probably another $1 million question, but I've always, it's something that I can't wrap my head around.
Yeah. So I, to be honest, I haven't put a ton of thought into that specific question, although I do think it's fascinating. There's some interesting research from Scott Dindot's lab a few years ago, which suggests that it's not really about, neurons needing to control the levels of UBE3A through this mechanism per se. So yeah, I don't really understand why this might have evolved. It is pretty clear, though, that the imprinting mechanism is conserved in most mammalian brains. So it's something that's been with us for a long time, whether by accident or by kind of selective pressure, like it's doing something. It seems to be the case. I think like Stormy Chamberlain's work has really started to get into kind of how this is the case. And it just appears so that imprinting, like the methylation happens in all cells, so that this methylation event on the paternal chromosome that, the lack of methylation on the paternal chromosome that allows this long non-coding RNA that encodes the UBE3A antisense, this thing that's interfering with the UBE3A transcript on the paternal allele. This long non-coding RNA is expressed by all cells. But what's interesting in neurons is it gets to read all the way through to the UBE3A region. And so it seems like there's some really key kind of bits of DNA in that intervening space in neurons that are just a little bit different. And there's something really curious going on that allows that read through all the way to UBE3A and to enable the silencing in a specific way. So I'm really always staying tuned to Stormy's great work.
Yeah, that's really
and how that
interesting
might be taking place.
Yeah, that's definitely something I'll look into as well. Well, you had started to talk about kind of neuron development and UBE3A, and how does it play a role in neurons developing as a patient gets older or even at that young age?
Yeah, so it's a great question, and I think largely it's really still a mystery exactly, what UBE3A is doing in a developing cell. So it's been linked to a lot of processes, like we've done some work in Ben's lab looking at neuronal growth. So we know the axonal compartment is really, really affected by loss of maternal UBE3A. So the axons that support all the presynaptic structures, they're just not as big, and they're potentially have a delayed myelination. I think this is coming out of some of the structural MRI imaging of patients with Angelman syndrome. So that's one really important developmental milestone, right? Like kind of growing the axonal compartment and myelinating it so that you can get really fast signal transduction. So that seems to be affected. You also seem, at least from the mouse studies, to have a lot of synaptic problems. So it seems like synapses may not develop appropriately and may not respond to experience the same way that that they would in a neurotypical scenario. So this is obviously going to affect the way learning happens. And there have been some nice studies linking UBE3A. I mentioned like the effects in 5 biology. So to different synaptic effectors like in the synapse. And we just know from just kind of looking into the brain and seeing where UBE3A is in neurons, that it's kind of poised at really important spots in the neuron to be doing a lot of things developmentally. So it's in, synapses, it's in the nucleus, it's in mitochondria, which kind of make all the energy for the cell. So, energy metabolism, which is, neurons are like voraciously using energy, especially during development. I mean, that could kind of really derail things if when UBE3A is not there, depending on what its roles are. So I know that's a lot to unpack, but it's really the case that there really could be kind of a domino effect when you take UBE3A out, and kind of problems radiating out in a multitude of pathways within the cell.
Yeah, well, I think it's really interesting because I think if you ask a lot of Angelman parents or, what was the takeaway they got from UBE3A, and they'd be like, oh, it's a housekeeper, it's just cleaning up these things. And I think that, there's, like you're saying, there's so many other things that could be involved with in how the neurons are physically. So that's really fascinating. And that kind of brings me to my next question. So I've heard the term, so dendritic spines is a term that I have heard, and I'm not a neurologist, but it seems, correct me if I'm wrong, so there's less dendritic spines in neurons with Angelman than a typical cell. And what do those do exactly?
That's probably generically true, that observation. I mean, with the caveat being, all things being equal, researchers, especially in the mouse models, as hard as we've looked to kind of document these spine changes, we've still only kind of scratched the surface of all sort of the territory in the brain to cover. But what they are, if you can imagine, so a neuron is a polarized cell. It's a really it kind of imagine like a tree where it's got kind of a trunk and an arbor, and that's kind of what the dendrites are like. And this is the part of the neuron that can receive information. And then it's got a root system, and that's kind of, think of those as kind of the axonal arbor, the roots, and that's what it's sending information through. So let's just focus on the trunk and the arbor and the leaves. So the spines are probably most similar to, they're like little outpouchings of the membrane, and they kind of will assume shapes like almost like little thorns or mushrooms sometimes, or kind of just like little stubs, and they're just decorating the neuron. And a single neuron can have thousands of these. And at each point, there's an opportunity for contact to receive neurotransmitter information from another cell, or like a cell that's presynaptic to it. And so what these little outpouchings of membrane do, they're very specialized. So they have lots of receptors for these neurotransmitters, and they make the transmission of information from the neurotransmitter into the cell like a lot more efficient. So they can transduce that signal into basically electrical activity really efficiently, much more efficiently than if it was just kind of flat and not specialized. And what's cool about them is that they can be really plastic. So you may have heard of a term called synaptic plasticity. So at least from the dendritic spine side of things, these spines can actually change really rapidly sometimes. They're kind of shape, their morphology, and also their neurochemical composition. So they can change a shape to more efficiently communicate a synaptic signal that they receive. They can traffic more receptors up to their cell surface to strengthen a signal. They also can go the other way. They can become more weak. So they can, undergo opposite changes that make that communication a little less efficacious and a little less efficient. And all of this ability to change is really important because it's thought to be what subserves, learning, and an ability to encode experiences in the brain. So we know UBE3A is there. I don't know if we know exactly what all UBE3A is doing in terms of synaptic plasticity, but it's certainly poised to play a pretty important role in that process. And, dendritic spines are just one synaptic location in which UBE3A might really be important.
Gotcha. That makes sense. So obviously, in these neurons with Angelman, UBE3A is lacking, and these neurons may be more immature and less fully developed, or less, not communicating as well. Do we know, so let's say, and we'll talk about these new therapies, but if we are able to give UBE3A back in some degree, do we know if those neurons will kind of just start functioning normally and be able to learn and communicate at the same level, or is it a long process, or are you kind of waiting for new mature neurons to be developed?
Yeah, so one interesting aspect of neurodevelopment is most of the neurons that you'll have in your brain are developed before the end of the second trimester in utero. And so with a few interesting exceptions, you're not really going to make a lot of new neurons after you're born in your brain. But what's really interesting is those neurons can undergo a lot of, they're still developing. Just because there's not new ones being born, there's still tons of development and kind of sculpting of the connections between them. So synaptic connections, outgrowth of axons to connect the distant parts of your brain, of your body with your brain. Like, famously, you have neurons up here in your motor cortex that talk all the way to the neurons in your spinal cord in your lower back that control your legs. So we're talking in an adult, like that's a meter long. That's incredible that there's a connection that long in the brain. And so these connections, I think, are what, kind of restoring the restoration of UBE3A through a therapeutic could be acting on. And so a lot of research says that the earlier you intervene, before a lot of these connections, get sculpted and become kind of more permanent, so to speak, is, the better your chances for, affecting therapeutic benefit are going to be. And that kind of makes sense. But what's kind of interesting is that we just flat out don't know what the potential really is. And so the mice these are all from like mouse modeling experiments, these observations. Mm-hmm. I think this might be a general truism. It does seem like there's a lot of synaptic plasticity that can be recovered basically throughout life in a mouse, insofar as we've looked. So definitely in adult mice, we've been on the subject of synaptic plasticity. There's really important synapses in a part of the brain that's involved in learning and memory called the hippocampus. And putting UBE3A back into these cells in the hippocampus fully restores certain forms of their synaptic plasticity. So the idea might be that while you might not be able to recover everything, or at least the way it typically develops over the course of neurodevelopment, you might be able to restore plasticity in large part, and that plasticity could be leveraged by a circuit to find maybe a new way to do something and perhaps, just to put a finer point on it, to recover some kind of ability that wasn't fully developed in the first place. So, maybe it's a motor ability, maybe it's a cognitive ability for an individual receiving a treatment. And so gotcha. I think that remains to be seen what the potential for that really is.
Are you struggling to find a doctor who really understands your loved one with Angelman syndrome? As a parent of a child with AS, I know firsthand how frustrating that can be. That's why we have established ASF clinics by partnering with leading medical and research institutions to provide expert, comprehensive, and evidence-based medical care dedicated to individuals with Angelman syndrome. We know access to specialized care is essential for your loved one to thrive. We have 23 locations in the United States, 6 globally, and with many more to come. Find a clinic near you at angelman.org/clinics.
All right, so we had talked briefly about how on the paternal side of the 15th chromosome, UBE3A is imprinted or turned off. So how has that been a target for research and therapeutics?
Yeah, I think it's been recognized since people, started thinking about therapies for Angelman syndrome based on putting UBE3A back into neurons. It's really been an attractive target. And the idea is that, hey, it's kind of a dormant copy. And if you can just figure out how to get rid of that antisense transcript or stop the stop of that's caused by the antisense, then you can basically turn on an endogenous copy of UBE3A that, as best as anyone knows, will work just like the maternal copy that's lost. And so it's really attractive because you should be able to express it in neurons the way it's supposed to be expressed. So all the, it's the same promoter. It should be expressed at the same levels. You shouldn't really have a risk of overexpressing it in most cases. So those are all really attractive things. And I think that's why a lot of strategies are based on doing exactly that.
Yeah, it's definitely an exciting strategy. I'm always curious. So let's say you are reactivating that copy. How do we know how much UBE3A we need as a target? Because on the other side of it, and this is probably a whole nother episode, but so with Dupe 15, you have too many copies and too much UBE3A produced, and you obviously don't want to get to the excessive point. So do we know if there is like a sweet spot, or how would we even, figure that out?
Yeah, so we have some research tools that we can use in kind of, mouse modeling systems, maybe even cell cultures or patient-derived neuronal cultures, where we can test that question. I think the short answer, and I don't want to sound like a broken record today, is we don't exactly know, but it's the truth. But it is, like, as you pointed out, it's a super important question for these therapeutic approaches based on restoring UBE3 expression in the brain. And so let's first kind of break down a little bit, like, this idea of the percentage, because there's kind of like two facets of it. There's how many neurons have to be re-expressing UBE3A, so kind of the percentage of all neurons. And then if you just focus in on the cells themselves, like, do they need a full dose to do their job, or can they get by on 20%, 50%? And we don't really know the answer to either question, but there are some clues out there from individuals with mosaicism, mosaic Angelman syndrome. Mm-hmm. And it seems like, if you have kind of 10 or 15% of your cells possibly around that might be expressing kind of neurotypical UBE3A levels, that you have a less severe form of the syndrome. Mm-hmm. So more speech, less severe motor problems, as I understand it. And this is all based on the assumption that, this mosaicism that's been measured kind of in peripheral blood cells is kind of reflected in the brain to the same degree. And so we don't know the answer to that question, but it's actually pretty promising that there's a kind of a continuum of kind of development that is UBE3A dependent. So another way of putting it is, an optimistic viewpoint is you wouldn't have to perfectly re-express UBE3A to 100% levels in every neuron in the brain to start to recover neurological functions.
Gotcha. So you don't need to really get close to that barrier where you're you're tiptoeing the line where you're going too much. You could probably theoretically go a little bit slower and less and still see improvement in some regard.
It's possible, yeah. And with certain therapeutic options too, you might be able to sort of, tune the dose a little bit. With something that's more of a traditional gene therapy, it's more of a one-time dose, and there's kind of more of a challenge of kind of getting the level right on the first couple tries, maybe, let's say, on the way things look now. Whereas with something that is, maybe more like, what's in clinical trials right now, these ASO-based approaches, you might have the opportunity to more carefully dial in the best approach. Although I don't really know what kind of the dose response is yet from these clinical trials. So that'll be that in and of itself will be interesting to see. Like, do you really actually have an opportunity to kind of tune this in, or is it just really slippery, kind of like, hey, knocking down the antisense, you ramp up to whatever the full dose is in the neurons that the therapy reaches right away.
Gotcha.
But yeah,
that makes sense. Okay. Well, yeah. So you just brought up, so the other strategy then is kind of gene replacement therapy, which you guys have been working on in your lab. Mm-hmm. So how does that strategy work, and how are you accomplishing that?
Yeah, so it's a very straightforward strategy. And so some of the disadvantages I alluded to, right? Like let me just break down the basic approach. So we are taking, and a lot of gene therapy programs are based on this general approach that I'm about to describe. So we take a viral vector called an adeno-associated virus. It's AAV for short. And essentially what we're doing is we're taking it, we're kind of hijacking it for our purposes. So these viruses have properties where based on kind of their envelope that normally packages their own DNA. This envelope can help them get into cells and introduce their DNA to cells. And so what we do is we take out their DNA and we put in our DNA, which is going to be UBE3A DNA in this case. So we're going to hijack the virus and make it do something advantageous from an Angelman syndrome therapy perspective. And so what our challenge then is to engineer into this vector, this kind of DNA that we put inside of this virus, this viral capsid, is we have to have it govern the expression of UBE3A as best as we can, as close to the way a neuron would normally do it. It's not like the unsilencing approaches that we were talking about, where you stop the antisense and, okay, now this dormant allele gets to just do its thing the way it's supposed to do it. We have to actually introduce the kind of regulation of the gene expression. And this includes levels, and which is can be a challenge. And it also includes making all the different kind of flavors of the UBE3A protein. So these flavors are kind of called isoforms. They're subtly different sequences of the protein that may confer to it slightly different functions and kind of let it travel potentially to different parts of the neuron. So, maybe more towards synapses or more towards the nucleus, for instance. And so the latter part, we think we've got a good grasp on. So we think we've come up with a kind of a clever way to put into this AAV vector of a DNA sequence that can give us the flavors of UBE3A in the right proportions. And that's pretty critical and a good big accomplishment for us, because these isoforms have been really highly conserved throughout mammalian brain evolution. So they're there, and they've been maintained, from mice all the way up to humans throughout evolution. So we think they're important, even though we don't know exactly what they do yet. And so the next challenge, I think, is to for our vector design and control is to make sure, as best we can, we're not overexpressing the protein. And so, we're definitely working on that. And we're also trying to explore what are the consequences of overexpressing the protein in the first place. Because as you are probably well aware, and a lot of people who are going to watch this are probably well aware, there's sort of this kind of almost like an inverse syndrome called Dup15q syndrome, where you get too much of this 15q chromosomal region, right? Mm-hmm. And so we don't really know what the consequences of UBE3A expression by itself are. So Dup15q syndrome tells us if you get UBE3A and all the other genes in 15q along with it, it's not right. And we know UBE3A is important because we know maternal Dup15q is a much more severe form of the disorder. So, but what we don't know is if UBE3A needs help, so to speak, to kind of drive the syndrome. And our mouse modeling data tells us that maybe it does need help, which means maybe we have a little bit of a buffer for overexpression. And so just to quickly summarize, we really worked hard to make mice that overexpress exactly 1 or 2 copies of the UBE3A gene, which is the exact same genomic or genetic overdose that you would see in either interstitial Dup15q or isodicentric chromosome 15. So, and these mice, insofar as we can, translate our findings to human, they tend to be pretty neurologically intact. We're not seeing major problems with motor function, cognitive performance, or sociability insofar as we've looked. Interestingly, we do see, if we really challenge them with seizures, they've got a susceptibility to succumb to these seizures, to basically die from a repeated seizure insult, which is reminiscent of this phenomenon called SUDEP, or sudden unexpected death in epilepsy, which tends to affect some individuals with Dup15q syndrome. So I'm not, by no means do we think overexpression of UBE3A is a good thing, but we are learning that maybe we have a little more wiggle room than we thought, which might make the gene transfer approach more viable and allow us to kind of take advantage of some of the advantages that it holds, which would be, a one-time dose potentially to kind of carry you through critical periods of neurodevelopment. So it might really hold promise, but we're just going to have, to do a lot more work to see if we can tap into that promise.
Sure. And you guys have seen some pretty good results with your mouse models with that treatment, right?
Yeah, so it's really interesting. We have done, so basically the experiment we did is we took our Angelman syndrome model mice with it, so the gene transfer therapy, and we injected them right when they were born. And so it's got mouse and human development aren't exactly kind of, they're a little staggered. So mice are born a little bit more immature than human beings are. So you can kind of think of a newborn mouse as sort of like the end of the second trimester human fetus. And so you might be thinking like, oh, that's like way too early, especially considering, current diagnostic practices, and you'd be right. But you also do have to factor in there's a little bit of a lag for our viral vector protein to actually kind of get into neurons, unpackage, and start expressing the protein. So we think we're kind of more along the line. So that we think that happens about the time the mouse is about a week old, which is much more consistent with maybe a truly newborn infant, which is still early, but and a little idealistic. But we're kind of starting to get more in the ballpark. And when we do that with this approach, and so we're basically putting it into the cerebral spinal fluid that's inside the brain ventricles. We are getting really good re-expression of UBE3A in the cortex and the hippocampus, and that, which is a little limited. So we're not getting it really well down into the spinal cord. We're not getting it back into the cerebellum, some motor areas, but we are getting it in cortex and hippocampus, and we're seeing decent behavioral recovery when it comes to seizures. In fact, we're seeing excellent recovery when it comes to seizures. So a normal seizure challenge that renders an untreated Angelman syndrome mouse really epileptogenic is does not have that effect in our treated Angelman syndrome mouse. So that was really, really encouraging. We're also able to recover a lot of things that mice do typically. So they're kind of like daily living behaviors are like digging and building nests, and they do that much, much better. We don't, however, recover their baseline motor performance. So if we challenge them with a kind of a motor coordination task, they don't, especially the first trial, they actually do very poorly, just like the untreated AS mice. And we hypothesize that this is probably due to the limited reach of the virus to, parts of the brain that are more likely to control motor function. But what's really cool, and I think kind of harkens back to what we were talking about earlier with regard to synaptic plasticity, they do seem to have gained this kind of motor learning ability. So they're really bad when they do this motor coordination task at first, but they're able to kind of catch up. So if you keep, testing them on it, it's like they are able to kind of train up to basically a wild type level of performance. So this might be kind of a learning mechanism, a sort of motor learning plasticity that we've been able to reinstate. So you might imagine an area of, the brain where we did put UBE3A back really well, like motor cortex, might be maybe taking over and conferring to the mouse kind of a new tool to reacquire some motor learning. So we've got a long way to go. We need to
amazing though.
Yeah, it's been encouraging, but we need to try to intervene later to better model what a likely clinical scenario would be in kind of older kids, and see what that yields.
Yeah, gotcha. Yeah, where do you see your research kind of going forward, or what is it you want to look into? What's the next step?
Yeah, as far as the gene therapy project goes, we really just are working hard to make it more translatable. I'm still working preclinically in the mice right now, but kind of as I just alluded, we really need to see what the potential for behavioral recovery is when we intervene later, see what our options for accessing other parts of the brain and the central nervous system are when we intervene with different routes of administration. So, trying to go through the lateral ventricles is great, but we also recognize that intrathecal delivery is right now a more common approach in the clinic, and so we're looking to see how well we're able to reinstate UBE3 expression and hopefully recover behavioral performance when we give an intrathecal delivery virus. So those are just some kind of brass tacks things that are just really important foundational studies to do. And then kind of on the other front, we're continuing to work with our UBE3A overexpression models to really kind of push them and try to figure out what it is that what overexpressed UBE3A sort of needs to really drive DUB15Q pathophysiology. Because I think that'll really not only inform mechanistic understanding of that disorder, which is super important as well. It's another UBE3A related disorder, but it will also inform our gene therapy studies as well, and inform the potential for risk with overexpression due to that therapy.
Yeah. Well, that's super exciting. And it's always interesting to hear the updates from you guys. And that'll be cool to see going forward. Well, I think that's all I have, unless you had something else you wanted to come back to. But man, we really appreciate all the work you guys are doing. And I think I speak for every Angelman family. It just means the world to us that you guys are even involved in this space. And I know Dupe 15 families feel the same way. So hopefully people get some good refresher and information from our talk. But I really appreciate you coming on, and this has been really great.
My pleasure, Christian. And it's a great opportunity to get to do this with you and with you guys. And it's a real privilege to get to work on something so important every day. So I think that's great. It's really inspiring.
Yeah. Well, thanks again, Matt. I appreciate it.
You bet.
Thanks for listening to the ASF podcast. If you enjoyed this episode and would like to support us, please share it with others, post about it on social media, or leave a rating or review. And please don't forget to subscribe.